Hello, my name is Avery Dodge, and this is my Journal Club 2 presentation. I decided to do it on the study titled "The Gut Microbiota Contributes to the Pathogenesis of Anorexia Nervosa in Humans and Mice." I also included the citation to that study at the bottom of this slide. It will also be listed at the end on the references page. First off, I just wanted to give you guys some acronyms just because they're woven through the study and it can kind of get jumbled or hard to understand if you don't know them. So I decided to put the key ones here so you could refer to it while reading the study or going through the presentation. As for my bluff, my bottom line up front, why should we care? Why should we care about this field? Well, specific to anorexia nervosa, it is a serious mental health condition and eating disorder with a high morbidity and high mortality. Lifetime prevalence of anorexia nervosa in adults was 0.6%, and 0.9% for females and 0.3% for males. I'd like to draw attention to that for a second. As you can see, the percentage for females is 3 times that of the males. Evidence base for treatment of anorexia nervosa is also lacking. Less than half of the cases achieve complete remission. The aggregate mortality rate is also estimated to be 5.6% per decade, which is much higher than the general population. As for the question asked within this study, the study investigated whether a disrupted gut microbiota contributes to anorexia nervosa pathogenesis. So what is anorexia nervosa? Here I listed just some key characteristics of a patient diagnosed with anorexia nervosa, which includes significant and persistent reduction in food intake, leading to extremely low body weight, relentless pursuit of thinness, a distortion of body image, and intense fear of gaining weight, as well as extremely disturbed eating behavior. Alrighty, so getting into gut microbiota dysbiosis, I just wanted to define it a little bit here before we move into the study. Within the human gastrointestinal tract, there is a complex and dynamic population of microorganisms in which make up the gut microbiota. Intestinal bacteria exerts a large impact on the host during homeostasis and disease, playing a crucial role in maintaining immune and metabolic homeostasis and protecting against pathogens. Gut dysbiosis, or also called altered gut bacterial composition, has been associated with the pathogenesis of many diseases and infections. This image comes from another Nature Microbiology article in which explores the gut microbiome and anorexia nervosa. As we go through the image, we can see how a disrupted gut microbiota, which is shown in the box A, can affect the host through increased satiety and metabolic efficiency. The field of research of eating disorders is multidisciplinary and has evolved significantly over the past century. Research may examine genetic, biological, behavioral, psychological, and social factors. The image I included demonstrates the estimated share of people with eating disorders from 1990 to 2021. Only anorexia nervosa and bulimia nervosa were included. Each age group is included, with the ages 20 to 24 and 25 to 29 having the highest percentage of eating disorder prevalence. These values were acquired through representative surveys, medical data, and statistical modeling. As the incidence rate of eating disorders climbs, especially in those younger populations, the urgency for research and treatment options also increases. Prior to the study we are discussing today, there have been smaller studies conducted to portray gut dysbiosis present in anorexia nervosa patients. Mouse models of anorexia have also shown that changes in the gut microbiota are associated with changes in eating behavior and expression of hypothalamic neuropeptides. So what is the urgency of determining if a disrupted gut microbiota contributes to anorexia nervosa pathogenesis? Anorexia nervosa, as I mentioned prior, has a high morbidity and mortality rate as well as a lack of evidence base for treatment. As seen in this table incorporated by the NIH, adults with anorexia nervosa sought treatment the least when compared to bulimia nervosa and binge eating disorder. By better understanding anorexia nervosa, we can improve treatments to hopefully raise these percentages in the future. To finally start working into Fan et al., I decided to start with this amazing diagram that they included at the bottom of the study. It basically just encompasses the entirety of the study. I urge you guys to look at this. It's just another great way to kind of understand the study. It has pictures and kind of simpler terms. So if you guys are struggling with all of this information in the presentation, you can come back to this lovely diagram and hopefully gain some more understanding. If you want this diagram to view on your own, if you save the study as a PDF, it'll be right there towards the bottom. So in this study, the authors explored their hypothesis that a perturbed intestinal gut microbiota and serum metabolome contribute to the complex pathogenesis of anorexia nervosa. This study involves only women, as women are the primary demographic for people diagnosed with anorexia nervosa, as I hinted at earlier. Since ethnicity may influence gut microbiota, only Danish Caucasian women with anorexia nervosa cases were included. The anorexia nervosa patients were all diagnosed by an experienced consultant psychiatrist and met the DSM-5 criteria for the restricting or binge purging subtypes. These participants were recruited from 3 specialized centers in Denmark. Exclusion criteria for anorexia nervosa participants included antibiotic or antifungal treatment within the previous 3 months and any acute or chronic somatic diseases or infections. All of the anorexia nervosa patients were treated in specialized centers and interviewed by an experienced and specialized psychologist or psychiatrist at the start of their treatment. Exclusion criteria for age-matched healthy control women were BMI below 18.5 or weight above 25 kilograms, regular medication of any kind apart from birth control pills, and antibiotics within the last 3 months. Control participants were recruited via public advertisement and through direct contact to health staff, medical students, and their relatives. Participants with anorexia nervosa were either inpatients or outpatients. Inpatients remained in routine treatment in the specialized unit for somatic and psychological stabilization. Weight restoration of 2 to 3% per week was the goal of the treatment in the inpatient unit. Care was given by a multidisciplinary team in accordance with international guidelines. Individually customized meals were given under supervision at scheduled times. If not consumed, a supplemental nutrition drink was added either orally or by duodenal tube. If a patient did not reach 2% of weekly weight gain, the energy content of the daily menu was increased. As for outpatients, anorexia nervosa patients paid regular visits to outpatient units where care was given by a multidisciplinary team. They were given an individual diet plan, which had the same nutrient composition as inpatients. All outpatients were also enrolled in cognitive behavioral therapeutic treatment courses, just like inpatients. As for sample collection, blood samples were taken in the morning after an overnight fast, collected on ice, and processed to obtain serum and plasma. Stools were collected in kits for anorexia nervosa cases and for healthy controls at home and immediately frozen until they were transported on dry ice to the biobank for DNA extraction. As for germ-free mice participants, female germ-free Swiss Webster mice were bred and maintained in flexible film gnotobiotic isolators until the start of the experiments at the Department of the Experimental Medicine at the University of Copenhagen. The mice were fed an autoclave chow diet and water ad libitum under a 12 hours light and 12-hour dark cycle at constant temperature and humidity. Fecal samples from randomly selected subsets of 3 patients with anorexia nervosa and 3 healthy controls who were representative of cases and controls were used to colonize 6-week-old female germ-free littermates. The germ-free mice were given a total of 2 doses of their respective fecal slurries, one on day 1 and one on day 3. After, mice were singly housed and given 30% energy-restricted autoclave chow diet for 3 weeks. Mice were weighed every 5 days after the start of energy-restricting diet. At the end of the study, mice were anesthetized and blood from the vena cava was collected so that plasma could be collected and stored. Inguinal subcutaneous adipose tissue, the cecum, and the hypothalamus of each mouse were dissected and collected for quantitative PCR analysis. For the methods of data collection in the study, I made a diagram to convey the critical details. And please give me some grace if I mispronounce some of these softwares. Bacterial cell count was measured using a BD Fortessa LSR2 flow cytometer, and data was acquired using BD Faxtiva TM software and processed using R package flowcore. Metagenomic sequencing was performed on the ion proton sequencer system. Meteor v3.2 was used to construct the gene count table of microbiome data, and low-quality reads were filtered by AlienTremor. Gut metabolic modules were generated by R-Package Omixer RPM. Viral gut microbiota was profiled by MyCop, as this is the optimized method to call viruses directly from bulk metagenomic sequencing reads and compute relative abundance within the virome dataset. Mass Hunter software was used for all metabolomic data acquisition. 16S rRNA sequencing of stool samples was done on an Illumina desktop sequencer. Illumina caught my eye immediately when first reading through this study's methods, as we have learned about this sequencing technology in lecture. Illumina allowed for high throughput DNA sequencing and rapid and large-scale DNA analysis in this study. Getting into the results portion of this discussion, I will be starting with the phenotypes of women with anorexia nervosa and controls. Compared to healthy controls, women with anorexia nervosa were much leaner, has lower fasting serum concentrations of glucose and insulin, higher insulin sensitivity, and lower serum C-reactive protein. Anorexia nervosa restricting type cases had higher values of serum insulin and lower insulin sensitivity than the binge purging subtype individuals. When comparing stool samples, there were no significant difference in bacterial cell counts. At the species level, the anorexia nervosa gut microbiota is characterized by higher beta diversity, which we can see in figure A. That burnt orange color is signified for anorexia nervosa cases, whereas that blue-purple color is for the healthy controls, and that is pretty consistent within the figures of this study. Within the anorexia nervosa subgroups, the binge purge subtype had a more heterogeneous bacterial community at the species level than the restricting subtype, which we can see in figure B. In figure C, we can see the species that were significantly different in abundance distribution between anorexia nervosa and healthy controls. Among the depleted species in anorexia nervosa cases were Rosaburia intestinalis and Rosaburia annulinvorans. These species are species that have had a high capacity for digesting plant polysaccharides and are considered to be part of the health-related gut microbiota. The authors investigated numerical covariations between the absolute abundance of bacteria at genus and species levels and bioclinical variables in the combined healthy control and anorexia nervosa cohort. They used a linear regression model, adjusting for confounders including age, smoking, and multiple drug intake. Interestingly, some bacterial taxa were linked to eating disorder scores and psychological conditions. It was found that Clostridium species were positively correlated with eating disorder scores, as we can see in figure D, indicating a potential role of these species in the regulation of eating behavior and neuropsychiatric symptoms. Absolute abundance of Lactococcus acidophilus and Faecobacterium prausnitzii were related to a score for interpersonal alienation. The absolute abundance of Parasitariella correlated positively with body dissatisfaction. The absolute abundance of Bifidobacterium correlated with a marker of perfectionism. Despite a similar absolute abundance of Brachyspira and anorexia nervosa and healthy control, This genus was positively correlated with markers for drive for thinness and anorexia nervosa. There was a higher viral richness and diversity observed in anorexia nervosa fecal samples when compared to the healthy controls, as we can see in figures A and B. As shown by figure C, 31 viral species that were enriched or decreased in anorexia nervosa were identified, and 25 out of 30 increased viral species were Lactococcus phages with known Lactococcus lactis hosts. These bacteria have been extensively used in the production of fermented food products. Analysis of viral bacterial correlations within the anorexia nervosa and healthy control gut microbiota revealed a decrease in the number of viral bacterial interactions in anorexia nervosa, as we can see in figure D. This was primarily driven by weakened interactions between viral species and short-chain fatty acid bacterial producers. When comparing the number of viral bacterial correlations in anorexia nervosa, restricting and binge purging gut microbiota, a reduction in the number and a much lower ratio of positive correlations was found in the restricting subtype. These results suggest that the gut microbiota in the anorexia nervosa restricting subtype has weakened gut viral bacterial interactions. Using gut metabolic modules and gut-brain modules to predict gut bacterial functional potentials, 159 modules were identified. The abundance of gut-brain modules for serotonin biosynthesis and the degradation of glutamate and tryptophan, which are metabolites with effects on mood and appetite, were found to be enriched in anorexia nervosa cases, as we can see in orange in figure A. On the other hand, the abundance of glutamate synthesis 2 and vitamin K2 pathways were higher in healthy controls, as seen in figure A in the blue color. Looking toward figure B, the authors found that serotonin synthesis and glutamate degradation pathways were inversely correlated with circulating concentrations of glucose and insulin or insulin sensitivity. There were no differences in gut metabolic modules found between the two groups. Bacterial genomes may have structural variants that potentially interfere with functional genes impacting the interplay between microbes and their hosts. Because of this, differences in the presence of or abundance of structural variants between otherwise identical bacterial strains may reveal critical phenotypic and functional differences. Anorexia nervosa and healthy control samples were significantly different in the beta diversity of structural variant composition, collectively suggesting that the composition of bacterial structural variants differs between the two groups. Exploring relationships between gut bacterial structural variants and markers of eating behavior, it was found that in anorexia nervosa cases, bacterial structural variants were significantly associated with eating disorder scores, as seen in figure D. The authors give an example of this using figure E, where it is shown that in the association analysis between structural variants in the B uniformis genome and host eating scores, 10 kpp deletion was directly associated with markers of bulimia and self-denial. This indicated that the deletion structural variant may be involved in the regulation of eating disorder and psychological traits in anorexia nervosa. B uniformis encodes a thiamine monophosphate kinase, see figure F, which is the distal enzyme involved in the thiamine biosynthesis pathway. Thiamine deficiency has been associated with mental health, including memory loss, anxiety, depression, irritability, insomnia, appetite loss, and gastrointestinal complaints. About 1/3 of anorexia nervosa cases suffer from thiamine deficiency. Anorexia nervosa cases lacking this bacterial genomic region were found to have higher scores for bulimia and self-denial, as we can see in figure G. The researchers performed untargeted metabolomics profiling of serum from anorexia nervosa cases and controls. This revealed a serum metabolome profile of 28 polar metabolites and 35 microbiota-related metabolites, which was significantly different between anorexia nervosa and healthy controls, as shown in figure A, but only slightly altered between the two anorexia nervosa subtypes, as seen in figure B. Serum concentrations of primary and secondary bile acids were higher in anorexia nervosa cases, indicating a potential role of the gut microbiota in anorexia nervosa related changes in secondary bile acid synthesis and metabolism and satiety regulation. Serum concentrations of indole-3 acetic acid and indole-3 propionic acid, true tryptophan metabolites, were higher in anorexia nervosa cases compared with healthy controls. Indole-3 propionic acid is associated with the secretion of glucagon-like peptide 1, which can stimulate satiety and slow gastric emptying. Bidirectional mediation models were constructed to explore the role of serum metabolites in the interaction between gut microbiota and host phenotypes. For direction 1, it was hypothesized that serum metabolites as variables mediate the causal effect of gut microbial features on host phenotypes. For direction 2, phenotypes were treated as mediators and alterations in metabolites as outcomes of changes in microbial features, as shown in Figure C. As previously mentioned and shown in Figure 1C, a drive for thinness outcome was identified for the anorexia nervosa microbiota enriched bacterial species C. parapetrificum, which was linked to changes in serum IPA levels, also enriched in the anorexia nervosa group. C. parapetrificum is a producer of multiple tryptophan catabolites, all of which are involved in regulating appetite and mental health, as shown in Figure E. And in another example, also shown in Figure E, indosyl sulfate was enriched in anorexia nervosa cases and was identified as a mediator of anorexia nervosa enriched Clostridium and the score for body dissatisfaction. Indosyl sulfate has been shown to induce anxiety or depression-like behaviors in human and animal models. Coming back to the germ-free mice of this study, after 21 days, germ-free mice transplanted with stools from anorexia nervosa cases showed a larger initial decrease in body weight and a slower weight gain over time compared with mice that received healthy control fecal transplantation, as shown in Figure A. The researchers performed hypothalamic gene expression analysis following the fecal transplantations. Anorexia nervosa transplanted and healthy control transplanted recipients differed in expression of multiple hypothalamic genes involved in controlling eating behavior and energy expenditure, as shown in Figure B. In anorexia nervosa fecal recipients, there was an increased expression of appetite suppressors and a receptor of serotonin. The authors also analyzed mRNA levels of genes encoding proteins regulating adipose tissue thermogenesis. This revealed abundances of UCP1, ELOVO3, and PCG1 alpha to be increased in the inguinal fat of anorexia nervosa transplanted mice, as seen in Figure C, indicated enhanced adipose tissue thermogenesis. 16S rRNA gene amplicon sequencing of human donor stools and germ-free mouse recipient fecal contents identified 85 overlapping amplicon sequence variants, as shown in Figure D, of which 45 altered amplicon sequence variants were transferred to recipients, as shown in Figure E. Amplicon sequencing variants and the expression of Browning genes, including UCP1, were strongly positively correlated, indicating amplicon sequencing variants to have a potential role in body weight loss or lower weight gain through enhanced adipose Browning. Overall, alterations in hypothalamic and adipose tissue gene expression and changes in body weight over time in the mice suggest that a disrupted gut microbiota in human anorexia nervosa may contribute to some of the elements in the complex pathogenesis of anorexia nervosa. Now to discuss the results relative to the bluff I mentioned earlier. Does a disrupted gut microbiota contribute to anorexia nervosa pathogenesis? Well, bacterial and viral components of the microbiome and the serum metabolome were altered in those with anorexia nervosa. Bacterial structural variants are different between anorexia nervosa cases and healthy controls, and germ-free mice transplanted with anorexia nervosa stools experienced more rapid initial weight loss and slower weight gain over time when compared to mice transplanted with healthy control stool. Bacterial metabolites may have an influence on anorexia nervosa behavior, affecting mood and appetite. When analyzing results from the germ-free mouse fecal transplantations, Anorexia nervosa transplanted mice, initial weight loss and slower weight gain over time was found to be associated with higher expression of appetite suppressor genes in the hypothalamus and higher expression of thermogenesis-related genes in adipose tissue. We don't, however, know exactly how the individual and combined effects of indole 3-propionic acid and myrcolic acid on energy metabolism, which future studies will need to explore. This study calls for further exploration of anorexia nervosa pathogenesis relative to the gut microbiota to ultimately understand the diagnosis for successful treatment in the future. As for the conclusion, this study ultimately supports the idea that a severely disrupted intestinal microbiota contributes to some of the stages in the pathogenesis of anorexia nervosa. And knowing this, future treatment of anorexia nervosa will be improved with the increase of understanding its complex pathogenesis. And as for the questions I have for you guys to kind of kickstart some discussion, I wrote question 1, how do you think better understanding how gut bacterial metabolites affect host health can further medical advances for treating eating disorders? Number 2, which figure from the study did you think was most important? Why? What did it contribute to the story? 3, what other sequencing techniques could have been utilized to sequence the 16S RNA from the stool samples, and why do you think Illumina was chosen? 4, what are the potential limitations of the study? Do you think the authors did well at minimizing these? And 5, what did you learn about anorexia nervosa? Did you know what it was prior to this presentation, and if so, what did you know? What did you learn that was most interesting to you? Last but not least, here is my references for this lovely presentation. I went ahead and highlighted the study that I discussed here in a lighter green color, so that way you guys can identify it easier. There's a lot of great studies and articles within these references. I found them very interesting, so I hope you guys do too. And with that, thank you very much for listening, and I look forward to discussion with you guys.